So Leah Mindorff will be presenting on evaluating the biodegradation of diluted bitumen in shallow groundwater systems using large-scale mesocosms. Okay, go ahead.
Yeah, sure. Let me just set up. Okay. Yeah, so my name's Leah, and I'm going to be talking about my master's project, where we evaluated biodegradation of diluted bitumen using some large-scale mesocosms. So to start off, I just wanted to clarify some of the terminology I'm going to be using when I talk about crude oils. So crude oils can be categorized several different ways, one of them being based on density and viscosity. So in that way, we have crude oils kind of laid out on a spectrum, going from light crude oils to heavy crude oils, and then all the way up to super dense, super viscous bitumen. And then we can also categorize the crude oils based on extraction method. So we have conventional methods, which would be like typical oil wells, and those yield conventional crude oils. And then we also have unconventional methods, which would be like the mining and the steam extraction that we see in the Alberta oil sands. So the Alberta oil sands produces bitumen, and it has chemical and physical properties that differ from conventional heavy crude oils. And the most obvious difference is that bitumen is so dense that it cannot flow. So in order to transport it using pipelines, we have to dilute it. And so what they do is they blend bitumen with a diluent, so which is just lighter hydrocarbon fractions. And it's called diluted bitumen or dilbit. And that's actually what's being transported through pipelines. And because there's such large volumes of dilbit being transported through pipelines across North America, there is, of course, the risk of an inland spill occurring. And so from previous studies on conventional crude oil spills, we know that one of the most effective and least invasive methods for remediating an oil spill site is through biodegradation. So there's many microorganisms that are naturally present in the environment that are adapted to degrading petroleum hydrocarbons. However, there are concerns that the unique physical and chemical properties of dilbit will cause it to behave differently than a conventional crude oil spill. And while the number of investigations into dilbit behavior in the environment is growing, there are very few studies looking at long-term dilbit biodegradation. And there's like pretty much a complete lack of studies investigating the fate of dilbit in shallow groundwater systems. So my master's project aimed to address those knowledge gaps by answering the following research questions. The first being, what is the impact of dilbit on a native soil microbial community? And second, how will dilbit biodegradation compare to biodegradation of a conventional crude oil? So we evaluated this using large-scale mesocosms, which acted as analogs to natural shallow groundwater systems. And so this mesocosm experiment was done in 2019. We had these open top columns that you can see here, which we filled with soil collected from the Mount St. Hilaire Nature Reserve, just outside of Montreal. And we ran the mesocosm experiment for 104 days at the lab here in Quebec City. So the test included a control tank, a dilbit treatment, and a conventional crude oil treatment. And we collected soil cores on a weekly basis for the first month, and then on a bi-weekly basis for the remainder of the experiment. And each soil core was divided into 3 sections. So we had a top, middle, and bottom section. And we did this because we wanted to see if the microbial response would differ with soil depth. So to assess microbial response, what we did was we extracted phospholipid fatty acids from the soils. And phospholipid fatty acids, or PLFAs for short, are basically the components that make up cell membranes, bacterial cell membranes. And so we use PLFAs as a biomarker, and they provide a sort of low resolution taxonomic profile of an active microbial community. In addition, the carbon isotopic content of the PLFAs can also be used to determine if oil is being biodegraded. So if dilbit is being degraded, for example, it means that microbes are using it as a carbon source. The carbon from the dilbit would then be integrated into the carbon in the microbe cell membrane. So the isotopic value of the dilbit would be reflected in the isotopic value of the PLFAs. So in our study, we looked at both stable carbon and radiocarbon isotopes, but I'm just going to be talking about the radiocarbon results today. So the results of the radiocarbon analysis basically confirmed that there was microbial uptake of both dilbit and conventional heavy crude. So here I have this graph. Basically on the y-axis, it shows the carbon-14 values. And then the x-axis is just showing the progression through the exposure. And then I have these two horizontal lines. I don't know if you can see my mouse. Maybe if I do that, can you see that? Okay, so this horizontal line basically represents the carbon-14 value of just the background soil organic carbon. And then all the way down here, we have the carbon-14 value of the two oils, so the dilbit and the conventional crude. So basically the results show that the triangles, the controls, were more positive or less negative, I guess, than the circles and squares, which represented the conventional crude and the dilbit samples. So this control value from day 104 and day 0, it's essentially unchanged. In comparison, we have our dilbit and conventional crude that get progressively more negative as the experiment goes on. And so we can see on day 104, there's a significant difference between the two oil-affected samples and the control, but we don't really see a significant difference between dilbit and conventional crude. So basically, we confirmed that dilbit and conventional crude biodegradation was occurring. However, the dominant carbon source was still by far the soil organic carbon. We only saw about between 10 to 29% of the carbon coming from the one of the two oils. So it's important to note, yes, biodegradation was occurring, but it was happening very slowly. So now that we had the radiocarbon data confirming that there was biodegradation, I wanted to get a more detailed look at the microbial community, so find out which bacteria were really thriving in a dilbit contaminated environment. So to do this, I extracted DNA from the mesocosm soil samples and amplified the 16S ribosomal RNA gene. And then those amplicons were sequenced and analyzed using bioinformatics. And the results of the sequencing are visualized here in this taxa bar plot. So we can see on the y-axis the relative abundance of each of the genera that were present in the samples. And then, so each of these columns represents a different treatment. Each of the rows represents the core depth going from the top to the bottom. And we can see as the exposure progressed in each of these samples, the red section gets bigger and bigger. So this red section represents Polariamonas, the bacteria Polariamonas. And we see in conventional crude and dilbit, it kind of shifts to becoming the dominant bacteria, but in the control, we don't see any change at all. So this is interesting because it makes it seem that Polariamonas is involved in the long-term degradation of dilbit and heavy conventional crude oil because it becomes the dominant taxa months after the actual spill. And this is actually consistent with other studies that have identified Polymonas as a petroleum hydrocarbon degrader. So to summarize everything, we ran a mesocosm experiment comparing biodegradation of dilbit and conventional heavy crude. Radiocarbon analysis confirmed that both of these petroleum products were being degraded, but that the soil carbon remained the dominant carbon source. Our DNA results showed that bacteria Polymonas thrived in this oil-contaminated environment. And overall, it seemed that the microbial response to dilbit and conventional crude was pretty similar. Our study is one of the first to investigate the potential for dilbit biodegradation in shallow groundwater systems. And we basically demonstrated that dilbit biodegradation by a native microbial community was possible. And in addition, it suggests that the biodegradation potential of dilbit is pretty similar to that of conventional heavy crude. So our findings can therefore be used to assess the environmental threat posed by different types of crude oils. And this in turn will inform the development of more effective spill response strategies.
Thanks, Leah. That's a wonderful presentation. Well, we have a couple minutes for questions for Leah.
Yes, I have a naive one, as usual. Thanks, Leah. Great presentation. And it's also a tongue exercise, Polymonas.
Yeah.
Even in French, it's impossible to say. For me, I mean, talking about after spill events. Is it, and I really naive, I don't know anything about that, but can if such an event arise, can we seed, I don't know how you say that in English, is it possible to inject kind of that kind of bacteria in the soil in order to accelerate this?
Yeah, so that is like a tool
digestion?
that a lot of people use and are investigating, as well as is using sort of bacteria that we know are able to degrade dilbit, and then bringing them, kind of like basically dumping them on a spill site. And there's pros and cons to it. It's when you introduce, you know, something new to a system, how much good is it going to do versus how much is it going to disrupt the existing community that's already there. Yeah, I don't know of any studies specifically that use Polymonas to sort of bioremediate an oil spill site, but there's definitely lots of other bacteria that they've tried with.
And just another sub question in that, is there any link between the temperature of those bacteria, of the activity of those bacteria with related to the temperature? I mean, if you are in Alberta,
yeah,
and it's minus 1,000 during winter, sorry, is it different than plus 85?
Yeah, for sure.
I'm talking
for sure.
in Celsius, obviously.
Yeah, your community is going to look, microbial community is going to look different, like at each season, at each different location. Polymonas, as the name would suggest, thrives more in like cooler temperature soils. Yeah.
Okay, thanks.
Oh, just one more question.
Just a quick one. Through that 104 days, is there more? Is it roughly equal amount of microbial activity going on all the way through, or do you know, or was there a depletion of other nutrient capacity within the soil to mean that the later ones had more or less microbial activity? So was that potentially other nutrients, other factors within the soil that could have been influencing those relative contributions?
I'm not sure exactly if I know what you mean, but I think what you're saying is, because it's just like a small system, are we just depleting all the nutrients out of it, and that's affecting the community?
They're not just the food, they are also a bunch of other, there's soil water, there's soil gases, there's soil phosphate, nitrate. All these things go into the microbial metabolic processes. And so it's not clear to me that the soil at the beginning has the same microbial supporting mechanisms as the soil at the end. And I'm not quite sure how you normalize for that.
Yeah, well, I think that's just like the flaw of running a lab experiment versus doing it in the real world. I'm not really sure.
Yeah, no, it's just an interesting one. I'm not sure. No, I don't have the answer, and microbes are far too complex for my poor brain. But
there's the control
yeah.
ball. There's a control right there in the middle. That's we don't.
The
we didn't
control's
add
undergoing the
much.
same problems. That depletion of some resources and enhancement of other resources, depending on how the microbial.
I mean, it's
population
also like
evolves
it's a
over the
soil
next 4
sample.
days.
It's not like a water sample. Water samples, you know, we get like the constant, you know, exchange from just like currents
Yeah.
and stuff. Like soil, I mean, yes, you're right, but like to a lesser extent, there's like less movement of nutrients.
Well, I mean, was there water put through this system?
Yes.
So that's going to also change the nutrient complement and
Yes.
the actual. So it's not clear to me that it's a level playing field all the way through.
Compared to.
So that compared to what so the microbial community there's clearly a change in microbial community.
Right.
But has that been influenced to some extent by the changes in soil composition associated with the experimental timeline?
And you're saying that these soil changes wouldn't occur in like a natural system?
They would certainly be different.
Well, yeah, of course.
So it's just interesting. I just wondered if you'd actually tried to control for that. That's all with say, nitrate analyses or
The control.
something like
Exactly.
that.
In the
No,
center,
it's not a
right?
control though.
That's the
Not
whole thing,
that
right?
kind of control. But it would be interesting to have that.
Yeah, it's
Anyway.
kind of like a transition from like a small scale like microcosm experiment to like the real world system. It's kind of like an in between of that.
Yeah, no, they're cool. No, it's good stuff. Really good stuff. I like it.